worry.
Okay, we have a few more minutes to finish this exercise. I just wanted to make a comment. So this is one of the fastest servers to do, well, the Swiss model server is one of the fastest to do protein structure prediction. There are others like the ITASR and what's the name of the other one that I want to say AlphaGo.
ITASR based in template is HHPret. ITASR.
ITASR can take some few days for you to get your results back. So Galaxy Web for protein modeling, yeah, the advantage of Galaxy is that you can have an entire project in the same workspace. It's something similar to what we've seen in the MPI bioinformatics toolkit. To study, at least in our, both of us work with very early proteins. And for our study, we usually, or at least I don't rely on the prediction of protein structure. And that would be more useful, I think, for if you want to model an interaction or a function. I think for early evolution modeling, I haven't used Galaxy Web. Have you used it, Carolina? I also think you don't do much prediction for your own research.
No, I haven't, but.
But yeah, I see how it can be very advantageous to have to do it.
So let me see the poll. Oh,
I think we are
one finished already.
Yeah.
Yeah, so I want you to mention also that we have some, in this part of this Swiss model, if you remember the scores that were an outcome of the TM score of the structural alignments, this Swiss model also gives some evaluation predictors of the model and parameters. And the first one is this one is GMQE, that if it is closer to one, is the closer to one, the better the model evaluation. And they also give another parameter that resides in each residue. So if each residue of the structural template and our target sequence has a probability assigned. This is the Q-mean value. But I also recommend you, because it also takes more time, but you can try definitely other tools like ITASR, for example, even if it takes more time. And for those that are in the part of the disorder prediction, I want to mention you something important, is because we are working here with UPRET. UPRET is one of the most widely used disorder predictors. Is like basic in some analysis, mainly when you are analyzing a big amount of sequences, and also complete proteomes, for example. But it's very interesting, or it's useful that we are doing this exercise because it's a predictor that is very fast. And in fact, all the predictors, the majority of times, they will output the disorder content of your protein in this kind of graphs. No, one is this kind of graphs, where every residue of your amino acid sequence will have a disorder probability assigned. So generally, those residues with higher, with a score higher than 0.5 are considered as disorder. So, and the ones that are below the 0.5 value of each of disorder prediction assignment are considered as order. So for example, this is our sequence, this is our input sequence, and those, this is the amino acid sequence positions of the residues. So all those residues that are higher, that has value higher than 0.5 will be considered a disorder. So if you are analyzing disorder content, is a very familiar way to see how can you interpret the data. Of course, this is not useful for high throughput analysis of disorder. Is more a visual, but you can also have some text files access to this disorder content predictions outputs, and you can download it in text format. And if you downloaded the text format of your results in UPRED, you will see each residue of amino acid in your sequence with a score. And every time that score for every residue amino acid is higher than 0.5, it means that it's highly possible that it's disorder. And yeah, so I think you are ready to finish, but yeah, also I present you a link to this PRO database that is the most famous database for protein disorder studies. And you can analyze all the experimental determined disorder that determines or that robust the disorder presence in a protein. So I think that's the majority has finished.
Yeah, I think we can go ahead and start the last part of this workshop. Thank you, Caro. And so my computer is going crazy now, but I hope you can hear me well and see me well. So this is the last part. We've been here a few hours now. And the idea of this last section is just to wrap up everything that we have done. You maybe will find out that you will not use all of what we have seen, but the idea of this workshop was not to make you experts on structural bioinformatics or in sequence analysis techniques. It was more like to get a bridge between these two types of analysis. So usually when you do structural bioinformatics, we usually focus on just analyzing structure prediction, and maybe some dynamics of our predictions or of our structures. And when we do sequence analysis, we usually do phylogenies and study evolution and mutation rate. And there are many bridges between protein structure and protein sequence. So what we wanted to show you is how to get from one to the other, how to extract information about sequence from structure and about structure from sequence. So that's what we are going to try to do, really wrapping up in this last section of the workshop. And we are going to go back to this slide where we have seen how a molecule can be used to compute a phylogeny. And here we saw that this phylogeny in particular was constructed using homologous sequences from a protein, which is myoglobin. And then the three-dimensional structure of these proteins was decorated with these colors to show how the sequence has changed. So actually this phylogeny is not constructed from protein structure. And protein structure is going to be a very useful tool. It's just that there are some things that we can't learn from structure. We need to learn them from sequence. So the relationship between protein sequence and protein structure goes like this. So the protein is encoded by the genome, which is at the DNA level. And if we have a mutation, this mutation can be translated into the amino acid sequence or can be muted if the mutation is a synonymous substitution. But all the other mutations will have an effect on the amino acid sequence. So the other types of mutations are non-synonymous substitution and insertions and deletions. Of them, insertion deletion processes are the ones that have the most impact on the amino acid chain. Then at the protein sequence level, at the primary level of the structure of a protein, these changes can be seen, but sometimes in structure, these changes are not so large. So even if we have a synonymous or non-synonymous substitution, the change can be very small. But if the mutation really changes some important part of our protein, then we will see that the stability of this protein will decrease or will change. Even it can increase. And this will have an impact on how we see the structure. Of course, from here we can learn that, for example, if we have a particular site that is very important for protein structure, this particular site will tend to have less mutations over time. And these mutations that are differentially happen in an amino acid sequence can tell us something about structure. So taking just this information about how proteins diverge, the first studies that were trying to understand if we can predict how much change in structure we will have from a change in sequence, we're taking everything that was deposited in the PDB and then comparing the sequences and the structures. And the idea was if we can predict the change in structure from the change in sequence, then probably we can say how divergent is a protein just by looking at its structure. And in the beginning, it appeared like there was a linear relationship between the change in sequence and the change in structure. And this was very interesting to find because once you get to the point where the mutations in a singular position are almost random because so much time has passed since the divergence of two proteins. So you have so much divergence that a position can become almost random in mutations. Then you could use protein structure instead of the sequence and see the distance between the divergence of these proteins just by looking at the structure. But then, now 20 years later, after this paper was published, the previous one, we now know that actually two protein structures that are homologous can have a large dissimilarity in sequence. So we can have really very similar structures with very different sequences. And also we can have very different sequences with very similar structures. So I'm saying this, but here this is showing that we can't really distinguish the behavior of a protein family, meaning proteins that are related by evolution, from the behavior of a non-homologous group of sequences. So it's really not that easy to trace back the divergence of two proteins using just protein structure. So this is a disadvantage of many methods that tried to do phylogeny based only in protein structure. But what we can do is really take the information that we learn from protein structure and then go back and compute phylogenies using sequence. Yeah, so you may have heard this term before, which is the twilight zone. This is the zone of similarity in sequence beyond of which, so less than 25% similarity, then the changes are almost random and we can't tell apart just using simple sequence analysis. We can't tell apart homologous from non-homologous. And this is also showing that the difference in structure is not different between homologous and non-homologous. So this idea that we can say if a protein is closer to another just by using structure cannot be used, but we can use the core of a protein fold to understand the evolution of different proteins. Here I am showing you one of the most diverse superfamilies of folds. These are the hop domains classified here in superfamily. And in this, I am showing you how the core of a protein domain is conserved, even though the external parts are not. So what things can happen to a fold during evolution? So there are changes that can happen in three-dimensional structure that are not going to change the linearity of the secondary structural elements in primary structure. So this is what I'm representing here. So here we have two structures, the retinal binding protein and the thrombin inhibitor. These are two homologs. And here we see something that is called a strand invasion. So the linear sequence of these two proteins is still the same, but in three-dimensional structure, what we are going to see is that a strand has invaded the place of the other strand in the homologous protein. So this is a higher order change that can be detected by structure superimposition methods, but cannot be detected in sequence alignment methods so easily, at least. Another type of change that is somewhat frequent during evolution is this is really going to affect the linearity of the sequence. And these are called circular permutation. This means that in the combination of elements of a protein are going to be permuted. So whatever was in the amino terminal part of a protein is going to become the carboxylo terminal part of the homolog. And these things happen during evolution. They were thought to be very infrequent, but we have seen many more examples over time as the technology to superimpose structures has become widespread. The last thing that can really hinder our understanding of protein evolution is the ability of proteins to move. So if we are studying a protein that has two different conformations, and then we superimpose even the same protein in two different conformations, we will have a large change. So that's one of the reasons why it's not the best option to use protein structure comparisons to compute directly a phylogeny, because even the same protein will have very large distances in a superimposition if it suffered just a change in conformation. But in sequence, this is not going to perturb the sequence of our protein, so we still can learn about the evolution using sequence of these proteins. Now, if you do encounter proteins that have two different conformations, let's say that one of the homologous is bound to its ligand and the other is not, you can still superimpose your proteins using what is called flexible alignment algorithms. And of these, there are two very famous, FATCAT and Rapido. And FATCAT is really going to change the paradigm of rigid body transformations, whereas Rapido, it's going to take the protein and it's going to break it in pieces such that you can superimpose the two body or rigid body fragments that match your target. So of these, I would prefer to use Rapido, but both methods are very useful. So in this last section, what I want to say is, let's go back from structure comparison to sequence analysis. So the idea of the last exercise is just take a structure superimposition and then understand what is happening to these proteins in sequence during evolution. So from a structure comparison, we can go back to multiple sequence analysis, and from this multiple sequence analysis, compute conservation and covariation. So this is my last section. That's why I put here my acknowledgements, but let's, so these are my funding agencies, but let's go back to the GitHub page and let's do the last exercise. And I think one of the questions before was about comparing more than two protein structures. So to do that, we need what is called a multiple structure superimposition algorithm. And in the Wikipedia page, there's a link in one of the exercises. There you have a list, a very good list of all of the methods that are available, well, many of them at least. And there you can find many methods to do multiple structure superimposition. Here I'm just showing you one of them, which is of very high quality, but the server is not friendly to the use of many users. So if you find that the server of Matras is too slow, it's because it can't really handle very well many queries at the same time.